Hi, good morning and good afternoon, everyone. Welcome to today's webinar titled Cracking Cellular Metabolism. I'm Andrea Klein of Labroots, and I'll be your moderator for today's event. This educational web seminar is being sponsored by Agilent. Before we begin, I'd like to remind everyone that this event is intended to be interactive, so we encourage you to participate by submitting as many questions as you would like during the presentation. To do this, you just type them into the Ask a Question box and click Send. It's helpful if you address each question to a specific speaker. We will be answering as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, you can also click on the Ask a Question box located on that far left of your screen. I'll now introduce today's speakers. They're both with Agilent Technologies. We have Dr. Mark Sartain, LC-MS Application Scientist, and Dr. Genevieve Van de Bittner, Scientist at Agilent Research Laboratories. For a complete biography on our speakers, you can visit the Biography tab at the top of your screen. But Dr. Van de Bittner, I'll now turn it over to you to begin.
Welcome and good morning. This morning's webinar will cover a new comprehensive workflow for cracking cellular metabolism, from sample prep to data collection and analysis. The first portion of the talk will introduce a new mammalian cell sample prep workflow that uses a room temperature metabolism quenching solution and solid phase extraction to provide metabolite and lipid fractions from individual cell samples. After introducing this new dual metabolite plus lipid sample prep workflow, Mark and I will share data from the application of this sample prep workflow to the analysis of the cellular response to drug treatment. This will be presented in the context of a broader and comprehensive sample prep, data collection, and data analysis workflow. Agilent has a long history of working with the metabolomics community dating back to the first International Metabolomics Society meeting in 2004. We've worked with the community to develop tools to facilitate metabolomics research. As you can see, the road from then to now includes many innovations in the metabolomics field, both software and hardware, from the Mass Profiler Professional Statistical Software Package to the Metabolomics DMRM Metabolite Data Analysis Database, to the Bravo Metabolomics Platform Automated Plasma Sample Preparation Workflow. The Agilent commitment to continued leadership and innovation in metabolomics should be clear from our extensive metabolomics portfolio. New additions to this extensive portfolio are increasing the number of comprehensive end-to-end workflows available, including the dual metabolite plus lipid cell workflow we will be presenting today. This comprehensive dual workflow takes advantage of a new cell sample preparation workflow, the latest Agilent Helix Z column, and the 6546 LC-MS system, as well as the latest data analysis software, including Lipid Annotator, Profinder 10, and Mass Profiler Professional 15. When combined, these sample prep, data collection, and data analysis solutions make a powerful, comprehensive workflow that allows for the cracking of cellular metabolism. The first portion of this webinar will focus on the new dual metabolite plus lipid sample prep workflow. One of our main goals for development of a dual metabolite plus lipid workflow for cell sample omics analyses was to make a workflow that could be automated. However, there are a few steps in the typical cell sample prep workflow that are difficult to automate. The first difficult to automate step is the initial step in the workflow, the quick metabolism quenching of the cells, wherein interconversion of metabolites via enzymatic activity is halted. Commonly, this step uses cold liquids, such as liquid nitrogen, which can be dangerous to handle manually, especially when one is in a hurry to quickly stop metabolism. These cold liquids are also incredibly hard to handle and store on an automated robotic system, which prevents automation of the workflow. After metabolism is quenched, the molecular components of the cells, such as proteins, metabolites, and lipids, are extracted, and for multi-omics analyses, different fractions of the cells are separated for more facile analysis via LC-MS. This fractionation is commonly accomplished via liquid-liquid separations, where polar metabolites favor an aqueous phase, proteins precipitate out, and lipids favor an organic phase. Although effective, liquid-liquid extractions are very tricky to precisely separate, both manually and when automated. To overcome the challenges of cold liquid metabolism quenching, we have developed a room temperature method for lysine mammalian cells and quickly quenching metabolism using trifluoroethanol. This room temperature quenching method improves workflow safety, reduces workflow complexity, and enables easy handling of the quenching solution by automated liquid handlers. To overcome the difficulty of separating the liquid layers of liquid-liquid separations, we have developed a solid phase extraction method. This extraction method utilizes the Captiva EMR lipid plate to separate polar metabolites and lipids for downstream intrasample multi-omics analyses. It is possible to automate these solid phase extraction workflow steps on the Bravo liquid handler. This diagram outlines the first major component of the new dual metabolite plus lipid cell sample prep workflow, Celllysis and metabolism quenching. Initially, cells must be removed from the media in which they are growing to isolate intracellular metabolites. After media removal, the cells can optionally be washed with buffers like PBS to further remove extracellular metabolites. Since cell washing can cause leakage of some metabolites and increases time before quenching, some prefer to skip the wash step. Next, the quenching and Celllysis solution is added. In this case, room temperature 50% trifluoroethanol in water. The cells are mixed with this solution to expedite the metabolism quenching process. So, how do we know that metabolism is actually quenched? We can add a carbon-13 labeled metabolite, like ubiquitously labeled carbon-13 glutamine, to the quenching buffer and monitor its conversion to downstream metabolites by cellular enzymes. Since carbon-13 labeled metabolites have greater mass than their endogenous counterparts, LC-MS analysis can be used to monitor these compounds in these samples. In this case, carbon-labeled glutamine is first converted into carbon-labeled glutamate before entering the TCA cycle. When an effective quenching reagent has been added to the cells, like 50% TFE, the conversion of carbon-labeled glutamine into downstream metabolites is stopped. This graph demonstrates this well. On the left, cell lysate from cells that have not had their metabolism quenched show a high conversion of labeled glutamine into labeled glutamate. Both a cold quench and our room temperature TFE quench prevent the conversion of labeled glutamine to labeled glutamate, indicating metabolism has been efficiently quenched. There is a small amount of measurable labeled glutamate in the quench samples. This is equivalent to the background level of labeled glutamate from control samples not containing cells. After cell lysis and quenching, the cell lysate is next separated into metabolite and lipid fractions. Separation of these fractions improves the LC-MS analysis of the samples by making it possible to target the separation of unique compound classes during liquid chromatography. This gives improved separations and increases the number of compound identifications. Furthermore, removal of lipids from the polar metabolite analysis helps increase column lifetime. To avoid the difficulty of hard-to-automate liquid-liquid separations, we have used the Captiva EMR lipid plate to complete a solid-phase separation of proteins, metabolites, and lipids. As shown in this diagram, the Captiva EMR plate contains a solid sorbent that specifically binds lipids containing acyl hydrocarbon chains and polar head groups. The sorbent does not have an affinity for metabolites that do not have hydrocarbon chains. We initially prepare the sample by adding an organic solvent to form a protein precipitate. Then, when we add the sample to the Captiva EMR lipid plate, the protein precipitate is caught on a filter above the EMR sorbent. The polar metabolites flow through the sorbent, and the soluble lipids are captured on the sorbent. Thus, the polar metabolites are collected in the plate flow-through and can be dried and reconstituted prior to LC-MS analysis. The lipids are then eluted from the Captiva EMR plate using a relatively hydrophobic elution solvent. Thus, metabolites and lipids are separated and collected from each individual sample. During the development of this workflow, some optimization was done to increase metabolite recovery. It was found that increasing the water content from 18 to 50% for a mixture of metabolite standards prior to passing them through the Captiva EMR lipid plate could increase the fraction of metabolite standards that made it through the plate. Improvements in recoveries were particularly good for bulkier, amphiphilic, and more hydrophobic molecules. When increasing the water content of the samples, we wanted to maintain protein precipitation as much as possible. To accomplish this, proteins are first precipitated by an organic solvent, then the water is added second to a final content of 50%. This provides significantly larger amounts of precipitate than when the organic solvent and water are added together, thus maintaining a protein precipitate level similar to that obtained by adding an organic solvent alone. You may now be wondering what the metabolite recoveries look like for this workflow when the cell sample matrix is present. To test this, we took frozen cell pellets, each containing 1 million cells, and prepared them using our dual metabolite plus lipid workflow. Then, to monitor recoveries, we added a carbon-13 labeled metabolite mixture to the samples before or after they were passed through the Captiva EMR lipid plate. The percent recovery for each carbon-13 labeled metabolite was measured by dividing the metabolite peak area from the pre-spike sample by the peak area from the post-spike sample. For this experiment, there were 6 pre-spiked and 6 post-spike samples, and we compared the dual metabolite plus lipid workflow to a liquid extraction workflow. Samples were analyzed by LCMS to find the carbon-13 labeled metabolite peaks. Here are the plotted recoveries for the carbon-13 labeled metabolites monitored with this experiment. On the left are results from the dual metabolite plus lipid workflow, and on the right are results from the liquid extraction workflow. Overall, the dual workflow provides excellent recoveries and also has a lower variability or a lower average standard deviation than the liquid extraction workflow. Thus, although the dual workflow can provide access to both metabolite and lipid fractions, it can also be used to obtain great and consistent recoveries for investigations focused on polar metabolites. Next, we looked at the lipid extraction performance for the dual metabolite plus lipid workflow. In this case, we had 6 sample replicates for both the dual workflow and a liquid-liquid extraction workflow. Lipids were extracted using both workflows and dried and reconstituted for LCMS analysis. The data files were then analyzed using Lipid Annotator, Profinder, and Mass Profiler Professional to find the annotated lipids extracted by each workflow. This is a heat map from Mass Profiler Professional that shows the approximately 200 lipids found in negative mode LCMS for the dual metabolite plus lipid workflow and the liquid-liquid extraction workflow. Note, the same lipids were found during data analysis across the 2 workflows. Additionally, 90% of the peaks found in the liquid-liquid method are present in the dual metabolite plus lipid method, with a peak abundance that is at least 50% of the peak abundance of the liquid-liquid extraction method. Overall, this indicates that the lipids normally found with liquid-liquid extraction are still found with the dual metabolite plus lipid workflow. Additionally, the dual workflow is also easier to perform manually and much easier to automate. As I mentioned in the beginning, the main reason for developing a room temperature metabolism quenching method and a solid phase extraction method for separating metabolites, lipids, and proteins was to make a metabolite plus lipid sample prep workflow for cells that was easily automated. While we haven't yet automated the cell lysis and quenching portion of this workflow, the use of room temperature TFE for cell lysis and metabolism quenching decreases the workflow complexity even when performed manually. We have automated on the Agilent Bravo liquid handler the separation and collection of metabolites and lipids using the Captiva EMR lipid solid phase extraction plate. This automation provides access to intrasample multi-omics analysis through hands-free sample prep. The automated method can be set up to allow processing of 8 to 96 samples at a time by using 1 to 12 columns of a 96-well plate. Now that we have covered the new dual metabolite plus lipid sample prep workflow, we will demonstrate use of this workflow in the context of a comprehensive, end-to-end sample prep, data collection, and data analysis workflow wherein we monitor the cellular response to drug treatment. To demonstrate our comprehensive end-to-end workflow, we used a well-known and well-studied cancer arthritis model wherein the K562 leukemia cancer cell line is treated with methotrexate, a drug used both as a cancer and arthritis therapeutic. Although well-studied, the mechanism of action for methotrexate is still incompletely understood. Recent studies have indicated that methotrexate causes modest decreases in relative abundances of multiple lipid classes and both relative increases and decreases in key metabolites. Thus, we aim to apply the dual metabolite plus lipid sample prep workflow in combination with LC-MS, data acquisition, and data analysis to the assessment of the impact of methotrexate treatment on K562 leukemia cells. The results were compared to a recently published study by Funk and colleagues. The comprehensive mammalian cell workflow we used to assess methotrexate treatment of K562 cells included a variety of Agilent Technologies solutions, including the described dual metabolite plus lipid sample prep workflow, sample analysis using Helix Z liquid chromatography, the 6546 LC Q-TOF, and analysis using Mass Center software, including Lipid Annotator, Profinder, and Mass Profiler Professional. The results of the comprehensive workflow are metabolite and lipid annotations, an analysis of whether there are significant changes to the metabolite or lipid upon methotrexate treatment, and an analysis of the specific metabolic pathways that are impacted by methotrexate. To complete the methotrexate treatment of K562 cells, we first divided the cells into 8 separate flasks and grew the cells over 2 splits to make 8 distinct biological replicates. On the day of treatment, cells from each biological replicate were split to a density of 4 × 10⁵ cells per milliliter. Half the replicates were treated with 1 micromolar methotrexate. The other half of the biological replicates were treated with a saline vehicle. After 24 hours of treatment, cells from each biological replicate were counted, and 4 aliquots, each containing 1 million cells, were transferred from each biological replicate to a microfuge tube for processing via the comprehensive end-to-end workflow. The methotrexate treatment very clearly prevented growth of the K562 cells, as cells treated with methotrexate had the same cell count after treatment as before treatment, while untreated cells roughly doubled over this same time period. Although growth was impacted by methotrexate, there did not appear to be changes in cell size or morphology. Now that I have described the biological experimental design for this comprehensive workflow proof of principle study, Mark will describe the sample prep, sample analysis, and data analysis of the samples.
Thank you, Genevieve, and thank you everyone again for joining us today. Okay, I will now pick up where Genevieve left off. She just described the sample study design in terms of cell culture and drug treatment. The idea here was to use those harvested cell samples and immediately perform the room temperature quenching, and then proceed with the automated dual metabolite plus lipid workflow just described. So we did this dual workflow. We collected the metabolites, dried, and stored at -80 to await analyses. We sequentially then eluted the lipids from the Captiva EMR plate, dried, and later reconstituted with appropriate LC-MS solvent for lipid analysis. And so I'd first like to focus on the lipid analysis with you, walk through with you that process, and discuss some of those results. This is just a high-level slide showing the major steps I'll describe for our targeted lipidomics workflow, and I'm just highlighting here some new features if you're already familiar with these products. We start with acquisition on our newest Q-TOF platform, which I'll introduce in the next slide. Then, really at the core of lipidomics analysis is our Lipid Annotator software for fast and comprehensive lipid annotation. We go on to feature extraction with Profinder, and we end with new specific support for lipidomics in our Mass Profiler professional software. Let's first talk more in depth about data acquisition. Now, last year, Agilent introduced a new LC Q-TOF, the 6546. So the 6546 delivers the industry-best combination of resolution and dynamic range designed to meet the challenges of metabolomics research. There is no longer a need to select high resolution or extended dynamic range mode. With the 6546, there is only one mode that provides these advantages simultaneously. The 6546 is also the first Q-TOF to have an isotopic fidelity specification, which is key to both metabolite identification and to qualitative flux analysis. Simply put, the 6546 is ideal for metabolomics analysis. Regarding the performance, on the right is a figure showing intrascan dynamic range from the same flow injection study. Here, the carbon-13 enriched E. coli spiked with carbon-13 C2 succinate with 3 technical replicates as overlaid. Despite the 4-order difference in intensities, mass accuracy and resolution are maintained and allow precise determination of both abundant and low-level peaks within the same spectrum. This is especially important when measuring complex matrices with high-throughput approaches such as flow injection analysis. So, TOF technology is an excellent choice for discovery metabolomics because it provides high resolution, high mass accuracy, and outstanding isotope ratio fidelity, and maintains its performance even as acquisition rate increases. To determine the ability of the 6546 to maintain mass accuracy and resolution at different acquisition rates, we assess the performance for different metabolites at acquisition rates from 1 to 50 Hz. The results shown here are for succinic acid. As is seen on the left, there is no significant change in performance. Being able to acquire results independent of acquisition rate accommodates high-throughput, high-speed chromatography, where chromatographic peak widths can be very narrow. So regarding chromatography, for this application, we chose the 1260 Infinity 2 Prime LC system. And we see several benefits with this system. The 1260 Flex pump has been designed to offer the 4-channel flexibility of a quaternary pump while matching the gradient and mixing performance of a binary pump. It also offers an 800 bar pressure limit, which met the requirements for the 2 types of applications that I'll discuss today, including separation of polar metabolites by helic and even separation of lipids by reverse phase chromatography. For lipid data acquisition, here we have a 19-minute reverse phase chromatography method that is well suited for plasma, tissues, and cells of eukaryotic origin. Shown are typical total ion chromatograms in positive and negative ion mode for the K562 cells we use in the study. Now, given the diversity of lipids, not surprisingly, these LC-MS chromatograms are very complex. But with advanced untargeted molecular feature finding software, we can pull out thousands of compounds from these data files, shown here as overlaid extracted compound chromatograms. An overarching goal of this particular LC-MS method was to capture as much of the lipidome as possible in terms of both chromatography and data acquisition. And so, with careful choice of mobile phases, with a single reverse phase-based method, we can cover most of the major polar and non-polar lipid classes present in a typical cellular lipid extract. Shown here is a positive ion mode chromatogram with overlaid compounds, where we are covering many lipid classes, from the smaller polar lysophospholipids to the late eluting and very hydrophobic triacylglycerols and cholesterol esters. Now, moving away from data acquisition and onto database creation, Agilent Mass Center Lipid Annotator is new software to support lipidomics. Unlike Profinder, it is provided free of charge to Agilent customers. Lipid Annotator supports both LC Q-TOF and IM Q-TOF MS-MS data. Lipid Annotator software uses multiple scoring algorithms and a modified theoretical lipid library to annotate MS-MS spectra based on in silico spectral matching. This theoretical library is a modified and extended version of Lipid Blast, containing 500,000 or so theoretical Q-TOF lipid spectra. Note that Splash lipidomics standard from Avanti has been added to the database in Lipid Annotator to support lipid class normalization in the downstream analysis. I think a really attractive feature is that the software is designed to not over-annotate lipid features, by providing only a level of structural information supported by the MS-MS spectra. The feature view shown here allows the user to get an overview of the results. The feature table contains all features with associated MS-MS spectra that were found in the project. There's a configurable feature plot where results can be displayed as abundance versus retention time, abundance versus MRZ, or MRZ versus retention time. This allows for a quality assessment of the annotation results. In the figure shown, the plot is configured to display retention time on the x-axis and MRZ on the y-axis. Only annotated lipids are shown, and they are colored by lipid class. The pie chart shows the number of lipids annotated for each lipid class. So the Match Details tab provides a more detailed view for examination of each lipid match. The lipid sum composition table contains information for each annotated lipid. Sum composition includes the lipid class, total number of carbons, and the total number of double bonds. The lipid constituent table provides possible R group matches for a given sum composition. The lipid constituent table includes the abundance percent score for each possible constituent, and if one constituent can be clearly distinguished, it will be reported as a dominant constituent. The mirror plot shows the observed MS-MS spectra on the top and the in silico database MS-MS spectra on the bottom for each selected lipid constituent. So let's step back to data acquisition for a moment and discuss a concept. Now, to create the AMRT database for the lipid profiling workflow, we need to acquire MS-MS data on the Q-TOF. As many of you know, comprehensive data-dependent LC-MS-MS analysis provides high-quality MS-MS. However, this mode can only select a limited number of precursors in a given time, so low abundant peaks may be missed. Iterative MS-MS automatically does multiple injections and excludes previously selected precursors on a rolling basis. In the example shown on the left, only 3 precursors are selected to simplify the graphic. On the first injection, the top 3 precursors are selected for MS-MS. Then these are excluded on the second injection, allowing lower abundant precursors to be selected. So how well does this work for lipids? The box plots of feature abundances correspond to annotated lipids from 3 sequential iterative MS-MS injections. As would be expected, we observed that the mean peak abundance of triggered lipid precursors decreased across the first 3 injections of lipid extract. So how does this compare to conventional auto MS-MS? The figure on the right shows a comparison between the 2 modes. With repeat injections in conventional auto MS-MS mode, only a small increase in annotated lipids is observed. In contrast, iterative MS-MS shows a significant increase in the number of annotated lipids. The number of injections done for iterative MS-MS depends on the complexity of the sample and the chromatography. For our cell-based study, we determined 5 iterative injections was sufficient. Okay, so here we have the results of a single pooled K562 lipid extract that represents the 32 samples. This single sample was analyzed by iterative MS-MS. These figures show lipid annotator results for positive and negative ion modes. For each ionization mode, 5 iterative MS-MS data files were acquired and analyzed. Lipid features are colored by lipid class corresponding to the pie charts, where the number of annotated lipids are shown as percentages. There are 271 lipids annotated across the 5 positive ion mode data files and 536 lipids in negative ion mode. While there are some lipid classes that ionize in both polarities, there are, of course, lipid classes that ionize in only one mode. This highlights the need in lipidomics to analyze samples in both modes. We were a bit surprised to see the larger number of annotations in negative ion mode compared to positive. This is in contrast to plasma and lit tissue extracts where we've seen a larger number observed in positive ion mode. I would speculate that the low number of neutral lipids in cells and the large diversity of phospholipids contribute to the differences we see here. The lipid annotator results were exported to a custom personal compound database and library known as a PCDL, containing accurate masses, spectra, and retention times, i.e. an AMRT database. Separate positive and negative ionization mode databases were created for subsequent analysis of MS1 data. The resulting PCDL databases can be examined and also edited in Agilent PCDL Manager. All right, so how do we leverage this database? Really, we have 2 workflows, untargeted and targeted. Now, with both workflows, the database is used to annotate MS1 lipid profiling data. Using MS1 data is a preferred approach as collecting data in this mode ensures adequate sampling of the chromatographic peak, even with fast chromatography. It also improves the quantitative results of lipidomics experiments. In the untargeted workflow, significant features are queued against the database to look for hits at the end of the process. For the sake of time today, we're going to walk through the results from the targeted workflow, shown on the right. And I like to think of this as a data-driven workflow, as the database is used as criteria for targeted feature extraction to only extract annotated lipids from MS1 data, again using the same chromatography that was used for database creation. In the end, those annotated results can be imported into Agilent Mass Profiler Professional software for chemometric analysis, as we'll show next. Now, as I mentioned, Profinder software was used to do targeted feature extraction on the MS1 datasets. The Profinder results were imported into Mass Profiler Professional for statistical analysis, where separate experiments were created for positive and negative ion modes. After normalization and baselining, the resulting PCA plots for both polarities were overall similar. Without filtering of entities, that is keeping all compounds, the results show tight clustering of the biological replicates within each condition, and demonstrated clear differences between MTX and vehicle. If you remember, there were 4 replicates each from 4 cell culture plates in each condition, which are indicated by symbols. We didn't see a separation here based on cell culture plate, and this indicates to us that really the drug treatment appeared to be the dominant variable responsible for the separation. One of the nice newer features of MPP allows creation of lipid matrix plots, or some would describe as heat maps. This type of plot shows differences between a methotrexate treatment and control, where each row is a lipid class, and each column is the average of 4 replicates from each cell culture plate. The abundance scale is red for high and blue for low. Clear differences were visually observed across lipid classes. In agreement with the findings of the paper we are referencing, ceramides were increased in the methotrexate-treated cells, while the reverse trend was observed for phosphatidylinositols. Now, for any particular lipid class, we can dig deeper and view the lipid matrix plot for the individual annotated lipids. Here is the lipid matrix plot for phosphatidylinositols, and in this case, all individual sample replicates are shown as columns, and the individual annotated PI lipids are shown in rows. In this case, we can see a clear trend where phosphatidylinositol lipids with relatively low number of carbons in their esterified acyl chains found to be decreased in methotrexate-treated cells. In MPP, it's useful to look at the data in different ways, and here we are showing a more traditional volcano plot using a t-test to pull apart differences between the methotrexate drug treatment versus the vehicle control. There are 63 significant features that pass the cutoff shown. We had to use a fairly modest fold change value of 1.5, and indeed the authors of the paper we are comparing against also found that lipid changes were fairly muted compared to some of the polar metabolite levels we'll discuss in a bit. Now, that paper also showed that a phosphatidylinositol with some composition 32,0 was the most significant lipid, and indeed we found the same lipid in our short list of lipids that decrease with MTX treatment, which is again in agreement with their findings. But with our approach in this instant, we are able to go beyond some composition annotation because there's MS spectral evidence at the lipid constituent level for the nature of the esterified fatty acyls. For a bird's eye view of the data, we can perform unsupervised hierarchical clustering. Here we are switching over to the positive ion mode experiment. We can already see that the biological replicas group together well with respect to drug treatment. But I think what is really interesting is to zoom in on a group of entities with notable differences. Here we see that a group of acylcarnitine lipids group together in an unsupervised manner and are increased with methotrexate treatment. Now, this lipid class was not reported by the authors, as acylcarnitines were not targeted with their methodology. So this new finding we are showing here seems to be the direct result of initially creating a database based in an unrestricted manner, where iterative MS data on the pooled sample was searched against a very large in silico library. Okay. I'd like to switch gears now and introduce the metabolite workflow. You can already see from this slide that many of the products are shared with the lipid workflow and were introduced a few slides back. In terms of software, some key differences here include the mechanism of compound annotation, and you will also see we can really begin to leverage biological pathways with Pathway Architect software. So, if you remember this slide, we had originally intended to show you today results from the dried metabolite extracts prepared with a dual workflow. However, due to COVID-19, we were not able to get into the lab to run and analyze the samples. Instead, I'd like to show you results from a small substitute dataset we had previously acquired. This dataset addresses the same biology we've already introduced, and I hope can serve the purpose of walking through the metabolomics data analysis workflow with you. Let me briefly describe this dataset, as there are some differences from the experiment we had originally planned, and also some limitations. First, the chromatography method is different than the highly optimized helix Z gradient that I'll show on the next slide. The dataset was acquired on the 6545 Q-TOF, in negative ion mode only, and with MS1 data only. The lack of MS data here means we can only go so far in the analysis, as we can't quite get to confident metabolite identification with this dataset. While we do have the same cell line and drug treatment, the concentration and duration of the treatment differed from that we used in the lipid experiment. There were only 3 biological replicates each, and a crude metabolite extraction protocol was used to extract metabolites, instead of the automated dual workflow we've been describing. However, despite these limitations, a coarse look at the total ion chromatograms shown on the right indicate that the replicates overlay nicely, and some subtle differences can already be observed from the TIC. As many of you know, good chromatography for polar metabolites has always been a challenging area. So in 2018, Agilent introduced a new helix Z column that is a novel zwitterionic phase and is designed to be stable at extended pH. It's also designed to be used with low buffer concentrations, which enhances MS signal. In addition to introducing the helix Z column, Agilent also introduced the Infinity Lab deactivator additive. This additive improves detection and peak shape for some metal-sensitive metabolites that can be problematic otherwise. Importantly for metabolomics, the ability to run at low and high pH allows optimal ionization efficiency for both positive and negative ion modes. The app note below references the best place to find these detailed methods and was optimized to separate challenging isomers of high interest. Moving now away from data acquisition onto software. So we have continued to enhance Mass Spectrometry Profinder feature finding software, which is critical to data analysis. Over many years now, Profinder has been optimized for both the accuracy of the feature extraction as well as the speed. Profinder uses a unique recursive analysis approach across the sample batch in order to minimize missing features. What I'd like to point out with this dataset is the new chromatowarping feature. One common problem is the alignment of features when chromatography is not completely stable, which is a common phenomenon with all helix columns. As you can see here, to address this issue, Profinder offers flexible chromatowarping to align the chromatograms prior to feature extraction. The results from Profinder were easily exported for subsequent analysis. For this batch, we kept only very high quality features, resulting in about 1,200 compounds for export. So after importing these compounds into MPP, here again we have the familiar PCA plot. And remember, we are only working with a negative ion mode dataset with limited replicates. Even so, in agreement with the lipid results, here we see a good separation of the methotrexate treated samples from the vehicle control. At this point, we annotated the compounds using the ID browser tool within MPP. Here we chose to search the Agilent Metlin PCDL. This PCDL is designed for LCMS analysis and contains approximately 80,000 compounds, including 11,800 metabolites with curated MS spectra. The Agilent Metlin PCDL is highly curated, which corrects errors found in public databases. The MS spectra are also highly curated to remove noise, ensuring only compound product ions are included, and correcting MRZ values to theoretical for maximum match quality. The Agilent Metlin PCDL can be user customized to add additional compounds, MS spectra, and/or retention times. We were able to assign putative annotations to 690 of the 1,188 compounds in the experiment. So let's get on to some more data analysis. We can create Venn diagrams in MPP. In this case, the results are interesting. There are 42 compounds present in the drug-treated sample, but not in the vehicle control. So from the putative annotations, what information can we find on the 42 compounds? Well, one of these was annotated as deoxyuridine monophosphate. Inspection of the Profinder project shown on the right confirms the absence and presence of this metabolite in the two groups. Now, DUNP was also reported by the paper we've been referencing as one of the top 4 most significant metabolites that increases with MTX treatment. It turns out there's a good mechanism for DUNP buildup explained by the authors. The enzyme dihydrofolate reductase is known to be inhibited by methotrexate. This would lead to the reduction of methylene tetrahydrofolate. Methylene tetrahydrofolate serves as a carbon donor in the conversion of DUNP to thymidine monophosphate. As for the remaining 41 compounds, more work is needed. We did not see a mass corresponding to the methotrexate drug itself, but this drug is known to be metabolized quickly in cells. Some of the other compounds could correspond to drug metabolites, including conjugates. For the remainder of the analysis, we chose to move forward with the intersection of the 1,146 compounds shared between the two groups, which was really as simple as clicking on the intersection of the diagram in MPP and moving forward. So proceeding with that list of features, here on the left, we perform unsupervised hierarchical clustering, and again the replicates group together closely. We also see a trend where we see a group of metabolites highly increased in methotrexate versus vehicle. On the right, a volcano plot shows 93 significant compounds with a fold change greater than 2. This also agrees with the paper, where there was a trend that overall metabolite changes were greater in magnitude than lipid differences. At this point, to move forward, we really need the MSMS information that we were lacking with the substitute dataset. We hope to get back in the lab as soon as possible and acquire this data. So if you're interested, please stay tuned regarding that progress. One of the prospects we are really excited about is the ability to correlate metabolite features with lipid features in a multi-omics analysis within MPP. Here is a proof of principle for the MPP correlation analysis between the 63 significant lipid features and 93 metabolite features. Clicking on any of these intersections brings up the correlation plot, where one can observe behavior across replicates. Remember, in the sample prep, we are extracting both metabolites and lipids from the same physical sample with the automated dual metabolite plus lipid workflow. So this intra-sample analysis would help to reduce variability compared to the common strategy where one has to split the sample for separate types of extractions. Okay, wrapping up the metabolite analysis, we finally introduced pathway analysis. So shown here is a cake pathway using the Pathway Architect module within MPP. And even with a list of very putative annotations, we see interesting changes in the pyrimidine metabolism pathway. Zooming in on a particular region, we see significant increases in DUMP, a metabolite we've already introduced. But we also see corresponding changes in the neighboring metabolite free deoxyuridine. Together, these results agree with the paper that there is a dysregulation of nucleotide metabolism. Okay, to summarize, Genevieve and I just presented a comprehensive end-to-end workflow from sample prep to data analysis to study cellular metabolism. In the first portion of the talk, Genevieve covered a novel method for room temperature metabolite quenching optimized for eukaryotic cells. The method is compatible with automation and enables sequential isolation of both metabolites and lipids from the same sample. Among other advantages, this workflow should enable direct intrasample multiomics analysis. Hopefully, I was then able to provide you with some familiarization of our key LC-MS hardware and software, including some new features that enabled an even more powerful analysis of molecular changes induced with drug treatment. But I want to end by mentioning that we just didn't have time to cover all the products in our metabolomics portfolio. There's much more beneath the surface to explore. For example, especially with the sample throughput capabilities, we could design large time-based experiments for qualitative flux analysis using VistaFlux software. We could also design complementary experiments with, of course, our GCMS offerings, or look at the cellular bioenergetics with our Seahorse products. So with that, Genevieve and I would really like to thank you for your time today and invite you to ask any questions you may have regarding the presentation.
All right, great. Thank you both for that very informative presentation. We're now going to start the Q&A portion of the webinar. If you still have a question you'd like to ask, you can still do so. Just click on that Ask a Question box that's located on the far left of your screen. Okay, let's get started. The first two questions, I think, are for Dr. Van de Bittner. How sticky is trifluoroethanol? Does it persist in LC-MS systems, and is there a washing protocol required to switch to a different method?
Yes, thanks, Andrea. There are a couple ways I'd like to respond to this particular question. I guess first I'll mention that in our method, the TFE is actually dried after at the end of the method, and the metabolites are reconstituted in a new solvent. So the TFE is actually not going on the LCMS system in our particular version of the method. That being said, there would actually be no issue at all with injecting TFE onto an LCMS system. It behaves very similarly to either ethanol or methanol. It's not sticky at all. It can easily be flushed off of the LC system. So that's not a particular concern. I will mention that I know maybe this question came up in thinking about some of the LCMS separation methods for LC chromatography. In this case, there are methods, and one of the ones I used to help develop the sample prep method was the reverse phase ion pairing method with tributylamine. And for some of these ion pairing methods, you do really want to ideally dedicate an LC to that particular ion pairing reagent. And you will need to clean the mass spec before switching to a new method. So there are cases where a switch like that might be necessary, but you wouldn't need to do it specifically for trifluoroethanol. If you want more information on cleaning the mass spec after using an ion pairing agent like TBA, there is a metabolomics CMRM database and method. Mark was heavily involved in its development, but there is a great cleaning procedure outlined in that method.
Okay, great. I've got one more for you, and then I'll move over to a couple for Dr. Sartain. Have you assessed the extraction efficiency of the Captiva plate for a structurally diverse metabolites? And is this data publicly available?
Sure, that's a great question. I think partly I'll answer this by going back to some of the data shared within this particular talk. In our optimization of the water content in particular, we did look at quite a diverse group of metabolites that let us know that increasing the water content of our samples before they go on the Captiva plate helps with their recovery from the plate. And so we did have CoAs as well as nucleic acids, organic acids, sugars, sugar phosphates. We're all amino acids, were all included in the metabolites that we looked at there. So I think we got pretty good coverage of different metabolite classes in that case. Moving to the recovery experiment, again, in this case, it's a slightly smaller group of metabolites we looked at that we actually had carbon-13 labels of. And those metabolites also included a variety of different classes from organic acids, nucleic acids, sugars, and amino acids. So we think there's pretty good coverage across metabolite classes for getting good recovery from the Captiva plate using our method. If you want to see these graphs in a published form, we did have a poster at ASMS 2020, so just a couple weeks ago. The poster number is MP391, if you're interested in looking at that, and then you can have the graphs available to view as needed.
Okay, great. Now I'll move over to one that I believe should be addressed by Dr. Sartain. Does Lipid Annotator accommodate all ions MS data from Q-TOFs without IM, and will that be supported?
Yeah, that's a great question. Thank you. So at this time, we're not really supporting IM for Q-TOF, all ions for Q-TOF without IM. The reason is we don't believe that gives the best quality data. What we see is that with all ions, you can lose the association of the fragment ions with the precursor. And as you know, many lipids share common fragment ions across many different lipids. And so it becomes sort of an analysis problem where you have a lot of ambiguity on how confident you are on your identified lipid. All ions is really useful for ion mobility, where the fragments are co-aligned with the precursors in terms of drift space, and there it has some good value. But I personally especially believe that in terms of just Q-TOF mode only, isolating the precursor, getting exact mass on the precursor and the fragments is really the best way to go for more confident annotation.
Okay, all right, great. One more for you, Dr. Sartain. With data acquisition, can you do blank subtraction with your iterative MS-MS?
Yeah, that's a great question. So yes, in theory, yes, you can do blank subtraction. And to do this, you would have your first injection in an iterative work list be simply a blank sample. And then most of these background molecules will be selected for MS-MS in that first injection. And those background precursors will then be placed on a rolling exclusion list and will not be picked in your next 3 to 5 injections of your relevant sample. So on the data analysis side, you would only load the iterative data files corresponding to your real samples, and you would leave out the blank. You would do this in Lipid Annotator, and you, in theory, get a clean analysis. But I would say that while you can do blank subtraction, there's some gotchas to be aware of. For instance, in negative mode, I see traces of ubiquitous and very common fatty acids, probably coming from mobile phases and surfaces. And these could be excluded from your analysis and your samples if you find them in the blank. You may not want to exclude those. The other danger is if you're in a hurry and you're running real samples and you're not monitoring carryover very well, carryover from your auto sample, for instance, you might see traces of the most abundant lipids in your blank from a previous sample. And then you might place those on exclusion list because you see them in your blank, and you miss those in your analysis workflow. So I often see, I think being overly sophisticated in your iterative MS setup with, say, blank subtraction is not always ideal. And I prefer just to put the results into Lipid Annotator and let the software sort out the results.
Mm-hmm. Okay. Going to go back over to one for Dr. Van de Bittner. For the quenching analysis with carbon-13 labeled glutamine, did you find any carbon-13 labeled metabolites downstream of glutamate in the TCA cycle?
Right, yeah, so this is an important question for answering whether or not you've really quenched the samples with the quenching reagents. In this case, for unquenched samples, we did find carbon-13 labeled succinate, malate, and aspartate from the TCA cycle. For the quenched samples, so either the cold quench method, which was done at minus 20 degrees Celsius, or the room temperature TFE quench samples, we did not actually find any of these carbon-13 labeled metabolites from the TCA cycle. So again, it looks like we were preventing the conversion of glutamine to glutamate and then also any conversion downstream of that. In addition to these carbon-labeled studies we did, we also tested ATP that was extracted from the cells. And we found that the ATP level that was extracted from using 50% TFE did not change if we left the samples out at room temperature for 24 hours. So this indicates that the ATP is very stable and that enzymes that would convert ATP into ADP or AMP are really inactive in room temperature 50% TFE.
Okay, excellent. Back over to Dr. Sartain. Do I have to follow specific chromatographic conditions for better results on the Lipid Annotator?
Yes, great question. No, you don't. You can use whatever chromatography method you have and you're happy with. You could use a 1-minute method, a 30-minute method. You could use a normal phase, reverse phase. It will come, as long as you have, good peaks coming out, then you can use whatever you want. Now, once you build your database in Lipid Annotator, and then you go back to that profiling method where you're requiring MS1 data, you will want to stick with that same chromatography method because that's where the retention times come from that you built in your database. Okay, so from that point on, you should stick with the same chromatography after you had the initial database built.
Okay, great. And I think we have time for at least one more, unless there's some more that are going to be coming in. And I think this can go to either of you. Is it best to do iterative MS-MS injections at the beginning and end of the run to have better RT drift correction?
Yeah, I would say that just depends on your chromatography and how much drift you're seeing. With reverse phase LCMS on lipids, gosh, I'm not seeing any drift at all. So I typically will put it at the beginning of the run. But that might be something you just want to monitor over time and see what the best strategy you might see there to see if you do have any drift. Then it would make sense to put it at the beginning and at the end to later have that data so you could compare.
Okay, great. And actually, it looks like we have time for at least one more. And I think this one goes to Dr. Sartain. Does analyte response still go down as the acquisition rate goes up on the newer 6546 Q-TOF? And doesn't that make really high acquisition rates impractical?
So I would say, yes, it's true that faster acquisition rates result in less dwell time for any particular spectrum and reduced ion statistics. So as acquisition rates go up, you'll naturally lose some sensitivity. So there probably is a practical upper limit on how fast you want to acquire with your experiment. But that plays a lot into how much sample you have to work with, the ions you're interested in, and how fast your chromatography is and how many data points across the peak you want. In some cases, you're going to want to go fast. And I think the point we're trying to make here is that with Q-TOF technology, you're not limited in how fast you can go in terms of mass resolution. You get the same mass resolution at 1 hertz or even faster, up to 50 hertz.
Okay, excellent. Well, thank you both again for your time today and sharing this important research. Do either of you have any final comments you'd like to make for the audience?
Yeah, I would say that we often get this question a lot since it wasn't asked. We often get the question of when is this new sample prep method going to be available and how do we get it? And we would just say that, we made a lot of progress in developing this method, but we currently do not have plans to release it as a product. But for those labs and you're interested in more details of the manual method and the automated on the Bravo, we'd really be happy to connect with you directly. And I also just want to mention that I said that we were anxious to get back into the lab and complete the metabolite analysis on the drug-treated cells. So if you're interested in that story, please stay tuned, and we hope to share with you those results in the future. And really want to thank everyone for joining us today, and thank you for your time. I hope you're excited as we are about this new sample prep method and its real potential in easing the pain of metabolomics research, where sample prep is often a bottleneck. So thank you, everybody.
Great, and thank you both again. And I'll echo your thanks to the audience for joining today and for submitting all of those interesting questions. For any questions that continue to come in during the on-demand period or that we didn't have time for today, they will be addressed by the speakers using the contact information that you provided at the time of registration. This webcast can be viewed on demand through June of 2021. But until next time, have a great day, and thanks again for joining.